Belesario, Group 15 with my fellow groupmate. So our topic is analysis and quantitative characteristic. The usual genetics analysis cannot be used on quantitative characters because one cannot be certain of the exact genotype of particular phenotype, and production as the exact phenotypic ratios are not possible. So the genetic analysis of quantitative traits therefore are quite different and have the following condition as a rule. Quantitative data consists of a numbers of measurement that constitute a sample. So, ideally, the sample of the population should be the same so that there will be no sampling errors. It is obvious that this cannot be the case always. So, this is the outline of our presentation. A is heritability estimation, B is genetic mapping, and C polygenic inheritance, D environmental influence, E is selection of breeding, F is statistical models. So, what is heritability estimation? The heritability estimation is the measurement of the proportion of variability in trait within a population that cannot be attributed to genetic factors. So the heritability estimate helps researchers understand the relative importance of genetics. This is the example of heritability estimation. The example is the researchers collect the data of the height of parents and their offspring considering both genetic and environment factor. Through analysis, they estimate the heritability of height. B is genetic mapping. Refers to the process of determining the location of genes on chromosomes. Today, the most efficient approach for gene mapping involves sequencing a genome and using computer programs to analyze the sequence to identify the location of genes. So there are the three types of mapping with those resources utilized. First is linkage mapping, second is pedigree, and then the third is polymorphic markers. So what is linkage mapping? Linkage mapping is a graph that shows how frequently during homologous chromosomes crossover. The more recombination of segregation occurs between two genetic markers, the farther apart they are thought to be.
What is pedigree? A pedigree is related to genetics is a chart that diagram conditions through generations of a family. Polymorphic markers. What is polymorphic markers? Polymorphic as related to genomic refers of the presence of two or more variant forms of a specific DNA sequence that can occur among different individuals or populations. The most common type of polymorphism involves relations at the single nucleotide, also called a single nucleotide polymorphism or SNP. Physical mapping, physical locations of the chromosomes relating more toward exact positions of gene elements. Cytogenic mapping, chromatic cells mapping, radiation hybrid mapping, restrictions mapping. What is cytogenic mapping? Cytogenic depict the locations of other or markers along chromosomes. Cytogenic maps are important in gene research as they related the genetic data and molecular sequence to the morphological features of chromosomes. Chromatic cells mapping. The somatic cells hybridization techniques for genes mapping depends on assembling panel of rodent human hybrid clones containing random subset of the human chromosome. Such panels should be as informative as possible and permit error directions and error corrections for each of the human genes in the various clones. Traditions hybrid mapping. It based on traditional includes chromosome breakage and analysis of chromosome segment retentions or loss using molecular markers. Restrictions mapping. Its method to use it to map an knowing segment of DNA by breaking into space and then identify the locations of the breakpoint. This method relies upon the use of routines called restrictions analysis which can cut or direct DNA molecules or short specific equation called restriction site. C. Comparative mapping involves establishing the relative genomic positions of or homologous genes. Pair is true specific such as the human mouse gene sequence database and DNA chips. Genome or genetic maps graphics restrictions of the relative positions of genes on the DNA sequence. DNA sequence especially the initial amino acid sequence of proteins as inspired by the ribosomes. However, complex imaging reactions in the cells can greatly modify the chemical compositions of nature positions. Database. A genetic database is one of the more set of genetic data, genes, product variants, phenotypes storage together with software to enable use of retrieve genetic data and extract information for the data. DNA chips are minimized microsystem based on the ability of DNA to spontaneously find and bind its complementary sequence in a high specific reliable reversible manner known as hybridization. Labels DNA molecules in a simple are analyzed at direct site on soil support.
Polygenic inheritance refers to the inheritance of traits governed by more than one genes. Generally, 3 or more genes govern inheritance of polygenic traits. Multiple independent genes have an additive or similar effect on single quantitative traits. Examples in human include height, skin color, eye color, intelligence. Environmental influence can affect the expression of genes. This can involve factors in the internal environment such as hormones or in the external environment such as effect of certain drugs. For example, barbiturates in acute, intermittent. Environmental influence like on behavior, gender role, sexual, differentiation, gender difference, integration, variation across culture, cultural influence, parent and peers, etc. Gender role. Our culture shapes our gender roles, expectation of how men and women are supposed to behave. Gender identity means how a person views himself or herself in terms of gender. Sexual differentiation. Sexual differentiation is not only biological but also psychological and social. However, genes and hormones play a very important role in defining gender, especially altering the brain, influencing gender difference, gender differentiation, integration. Men more than women express and behave in aggressive ways. This aggression gender gap appears in many cultures and at various age. In male, the nature of this aggression is physical. Variation across culture. Culture difference differ. Each culture develops norms, role for accepted and expected behavior. Men holding hands in Arabia is normal. Closer personal space, not American culture. Cultural influence. Humans have the ability to evolve culture. Culture is composed of behavior, ideas, attitude, values, and tradition shared by a group. Parent and peers. Parent and early experience. We have looked at how genes influence our development. Difference. What about the environment? How do our early experience, our family, our community, and culture affect this difference? Selection of breeding involves choosing parents with particular characteristics to breed together and produce offspring with more desirable characteristics. Humans have selectively bred plants and animals for thousands of years, including crop plants with better yields. Example of breeding goal for the next round of selection. Number 1 is definition of production system. Number 2, definition of breeding goal. Number 3, collection of information, phenotypes, family relationship, and genotypes. Number 4, determine selection criteria, genetic model, breeding value estimation. Number 5, selection and mating, predicting selection response, consequences of mating decision. Number 6, dissemination, structure of breeding program, crossbreeding. Number 7, evaluation, genetic improvement, genetic diversity. Statistical model is a mathematical representation or mathematical model of observed data. When data analysis apply various statistical model to the data they are investigating, they are able to understand and interpret the information more strategically.